We are delighted tonight to have Dr. Stephanie Hicks with us. Dr. Hicks is our first LSU alum to speak as part of this seminar series. We've had some faculty here and some people associated with LSU before, but never an alum, so this is nice. She received her BS in math from here at LSU in 2007. Went on to Rice, where you got an MS and a PhD, right, in statistics. And then in 2011, moved from there to Boston, where you, correct me if I'm wrong, you're at the Harvard School of Public Health and the Dana-Farber Cancer Research Institute, working in biostatistics and computational biology. And so she's going to talk to us tonight about why statistics matters in the analysis of genomics data.
Thank you. Thank you so much. I'm so excited to be here back on OSU's campus. I'll admit I haven't been back in like 5 years, and the campus has changed. There's now a bus. There was no bus when I was here, so that's pretty awesome. I ran the lake today. The lake looks the same, but generally I'm very excited to be back. So I want to thank everybody again for inviting me to talk. I want to share today with you some of my research interests and some of the things that I work on a day-to-day basis. As the title of my talk says, I'm going to discuss why statistics is so relevant and the major roles in the analysis of genomics data. So I work with researchers who sequence the genomes of all sorts of things. Cancer samples, people, animal, many different things. And the goal is to find genetic differences that can lead to physical differences. So in the case of cancer and normal, you want to find the genetic differences that are causing big differences between cancer and normal samples. Statistics is one way of doing that. So before I go any further, I want to take a minute and just illustrate how statistics can not only help in the analysis of genomics data, but also in the broader community of data analysis. And rather than just voicing my opinion, I thought it would be fun to start out with what other people are saying about statistics. So first, Hal Varian, he's a chief economist at Google and a professor at UC Berkeley. In 2009, he was quoted saying, "I keep saying that the sexy job in the next 10 years will be statisticians, and I'm not kidding." So if you don't get anything else from my talk, all I ask is you watch this really video. I'm going back what I'm going to tell you. That's all I do.
This issue of the availability of data is I was giving some career advice to students a few months ago, and I said, look, the critical thing to do is to be complementary, have a scarce talent or a scarce resource that's complementary to something that's ubiquitous and cheap. So this is like if left shoes are really cheap and you have a monopoly on right shoes, you're in a really good position. And the example here is what's getting ubiquitous and cheap? Data. What's scarce and expensive? The talent to be able to analyze that data and make it tell its story. So it's the analytic capability, which I think does involve computers, but ultimately involves the individual's understanding and talent and capability that is the dream job of the next decade. So I've been telling people that the really sexy job in the 2010s is to be a statistician, because they're the people that can tell a story. And everybody has data. And the problem is, how do you utilize that data more effectively? Because now we have these systems that collect everything. And the bottleneck is analyzing it. And there just aren't enough people to do it. And we've got a team, we've got at least 100 statisticians at Google. We've been building up, and they're at every aspect of our data, both on the search side and on the business side. It's a very critical role to play.
So what did you think of that? Because when I saw that in 2009 or 2010, I thought that was pretty cool. I mean, again, big data wasn't really big data yet. It was just the cusp of big data. But hearing someone like that say something like that was really interesting to me. So to summarize what he's saying is that it's now easier and cheaper to get data, but the bottleneck is being able to analyze that data and come up with ways to tease out the informative parts. So Bridget Schultz wrote an article in the Washington Post, I think in December, saying that women are flocking to statistics. She said the reason was that women tend to be drawn to the collaborative sciences that rely on teamwork and communication. And statistics is definitely one of those fields. I collaborate all the time with biology researchers and other statisticians in the department. You may have noticed the word data science in that title as well. I wanted to explain how that's related to statistics, as those two terms have been often used interchangeably. Hilary Mason is a chief data scientist at Bitly, and she views data science at the intersection of four disciplines: math and statistics, computer science, what she calls hacking, and engineering. But emphasizes that statistics is a major component of this. Another leader in our field is Jeff Leake from Johns Hopkins. He's a contributor to a really well-known blog called Simply Statistics, and wrote a blog post titled "The Key Word in Data Science is not data, it is science." So this echoes the importance of developing methods and the analysis of data rather than just focusing on buzzwords such as big data. So how are we doing in training students for statistics? Well, the number of students has risen steadily since 1997, or number of students in high school taking AP statistics courses has risen steadily since 1997. And for undergraduates, the American Statistical Association just put out a study 2 weeks ago, I believe, on February 3rd, reporting that statistics is the fastest growing undergraduate STEM degree since 2010 to 2013. So statistics is very relevant right now. Furthermore, for undergraduates and graduate students, the number of stats degrees has risen steadily since 2003. Interestingly, 2003 was the first year that the NCES, the National Center for Education Statistics, reported statistics as its own category. So before 2003, statistics wasn't even considered its own category. That's how new this field is. So to put this in perspective of my undergraduate career, since I'm an alum from here, I graduated high school in 2003. I completed 2 research experiences for undergraduates while I was at LSU. And then I graduated in 2007 with my bachelor's and started at Rice. So at the time of this transition, there was not a lot of hype about big data or data science. But there was a very big transition happening in the world of genetics and genomics, which is what drew me to biostatistics. So Elaine Martis from the University of Washington School of Medicine in St. Louis wrote an article and included this figure that explained this transition happening from around 2004 to 2006. All of a sudden, the amount of genomic sequencing data produced rapidly increased around 2004 to 2006. And this was due into a change in sequencing technology. I don't want to go into the details of the technology, but generally the shift was from what was called Sanger sequencing, which is a slow, labor-intensive process, to next generation sequencing, which is a faster and cheaper process, and currently what most people are using today. So around this time, the cost of sequencing also dramatically fell. So the amount of data, of genetic data, was growing rapidly, and the cost of sequencing was falling. This plot compares the price of sequencing. So this was the cost of genome sequencing in 2003, and this was the price in 2013. And it compares the price to Moore's law. This was almost an obligatory plot that I saw in every single presentation while I was in graduate school. Everybody compared the cost of sequencing to Moore's law. I mean, it was like obligatory. But it is interesting to see how much more dramatically the cost of sequencing fell off in the years after 2006, when there was so much data available. So what all this led to was researchers now had an enormous amount of data, and they had to explore it. And what they found was there were many challenges in how to analyze that data. So for the rest of my talk, I want to discuss 3 questions that researchers grappled with. First, how do we take this raw genomic data, this massive amount of raw genomic data, process it, and normalize it in a way that I can actually do something with it? How do I even just take it from the sequencer and get it on a computer? Now, once I get it on a computer, say I have groups of samples that I want to compare, cancer and normal samples. How do I find differences between these groups of samples? And then third one is, how do you find interesting genomic regions? So once you have the data and you've analyzed the data, how do you find regions in the genome that are informative or associated with a particular phenotype? So interesting genomic regions. So these are 3 basic questions that researchers struggled with for a really long time. So for the rest of the talk, I'm going to focus on these 3 questions, and I want to illustrate how statistics can help with these questions. I'm going to focus on mostly one type of genomic data, and that's DNA methylation. I'm not sure if you're familiar with it, but I'm going to give you a really high overview of it so you can follow along with the talk. Before I explain what DNA methylation is, here are 2 examples. In these 2 pictures, you have mice with the same genetic information, same genome. But what's different is the level of DNA methylation that are causing these different phenotypes. So in the left plot, for example, these 2 mice have the same genetic information, but the level of DNA methylation is different between the 2 mice, causing a kink in one of the tails. In the right plot, the change in DNA methylation controls the amount of yellow pigment in the mouse, even though they have the same genetic information. So again, DNA methylation is not genetic information, it's different. So DNA methylation is also involved in controlling gene expression, such as turning on and off genes, and it's also involved in the progression of cancer. So this is a really high overview of DNA methylation. Let's consider a double-stranded segment of DNA. DNA methylation is the process in which you add a methyl group to a cytosine base that's next to a guanine base. These sites are called CPG for shorthand, and they translate or stand for C-phosphatase G, and that is a cytosine and guanine base separated by a phosphate. And note that's different than the GC base pairing naturally. So interesting fact, in mammals, around 70 to 80% of CPG sites are methylated at any given point. That tells you just roughly how much methylation is going on in general. And this is an epigenetic modification that can be passed down or inherited through cell division. So I mentioned DNA methylation is involved in controlling gene expression. In general, when you have a transcription factor, it can bind to a promoter region of a gene, and then it will turn on the gene, essentially translating the DNA into RNA. And then when you have CPG sites in the promoter region, so these C-phosphatase G sites that are methylated, that have the methyl group attached to the cytosine base, this can prevent the transcription factor from binding to the promoter, and then that prevents the transcription of DNA into RNA, therefore silencing the gene because you can't turn on the gene. Okay? That's my biological understanding. Okay. So this is a common feature found in many human cancers, actually. So these are often referred, often related to tumor suppressors. The typical scenario is you have 2 groups of samples that you want to compare. So cases and controls, tumor and normal. You could have more than 2 groups, but just for now, consider just 2 groups. Here I'm going to label the blue samples as controls. There are 3 samples, and I'm trying 3 case samples. So say the controls are normal samples and the cases are cancer samples. Within each sample, we measure the proportion of cells that are methylated at a specific CpG site. So at one CpG site, we measure on average what is the proportion of cells that are methylated at that one site. So this is the quantitative measure ranging from 0 to 1. And the shading here represents the level of methylation, where light is not methylated, so close to 0, and dark is close to 100%. So we do this for hundreds of thousands of CpGs in the genome. So we don't just do it for one CpG, we measure it at many different locations. We can then compare the level of methylation in the cases and find CpG sites that are less informative or not that different in terms of methylation. So here's the methylation value around 3%, 2%, 0 are differentially methylated. So they have a different methylation status. So here, these CpGs in the case samples are around 80%, 50%, 95% compared to the controls, which would be like 1 to 1% and so forth. Does that make sense, the idea of differentially methylated regions, or differentially methylated CpGs? The goal is to find these CpGs that are differentially methylated between the phenotype that you're interested in. So the problem is, how do we find these CpGs that are differentially methylated? You could throw out many approaches, but some solutions that people have proposed are to consider just one CpG site and do what's called a statistical test or a hypothesis test that tests if there's a difference in the methylation status. And if you are familiar with any basic statistics, you probably have heard of a Ttest or an Ttest or a linear regression model. All of these have been proposed and all of these have been written on in an attempt to find differentially methylated CpGs. So this is an example of statistics put to use. It was really cool that this was actually able to find differentially methylated CpGs along the genome. So I want to give you an example. If you're not familiar with the Ttest, don't worry, I'm not going to put up an equation. I just want to give you some intuition of what's happening. If I consider one CpG site, so just one of those columns, and I plot the methylation level for the cases and the controls, I plot the control samples, the 3 control samples in blue, and the 3 case samples in red. And then a Ttest essentially calculates the average distance, or the difference between the case and the control. So you compute the average of the case and the average of the control, compute the difference of the two, and then a Ttest will tell you if that's statistically different enough. It's scaled by the variability, so if the CpGs are very variable, then the Ttest is less likely to believe that. If the methylation values are all not very variable and they're very tight and very different, then the Ttest will tell you, yes, that's definitely different. So rather than putting up an equation on the screen, like I said, I'm just going to provide a p-value. So it's essentially the result of a Ttest. All you need to know is that if a p-value is less than some threshold, that threshold is often called 0.05, then we would say this CpG is differentially methylated. Does that make sense? So let's consider a different CpG. We can go through this process again, compute the average methylation status of the cases and the average methylation status of the controls, calculate the difference, plug this into a Ttest, and it would say the p-value is 0.343, which is larger than 0.05. Therefore, we would say this CpG is not differentially methylated at a 0.05 alpha level. You're familiar with this. So that's the idea. Now, say we did this analysis for each CpG site in the entire genome, and then we ranked all the CpGs by how statistically different they are. Does that make sense? So we did this hundreds of thousands of times, and we found the top 600 CpGs. So these CpGs are very different from each other between cases and controls. Does that make sense? That's what I'm plotting along the rows. So I'm plotting the top 600 CpGs in the genome in this plot for 36 different DNA methylation samples. And these 36 DNA methylation samples correspond to 6 pure blood cell types. So natural killer cells are NK, CD8 T cells are the second batch, CD4 T cells, granular sites, B cells, and monocytes. That's what I'm plotting here. And this is formally called a heat map, if you're wanting to know what the name of the plot is. So the red colors represent the CpG sites that are methylated, so a DNA methylation status of close to 1, and the blue represents a DNA methylation status close to 0. So what you can see is if you say you take the first set, top 50 CpGs here, all of the CpGs and the first 5 cell types are methylated because they're all red. But in the last cell type, they're all blue, so these are not methylated. What this means is that pure cell types have a unique methylation profile that can be identified just using DNA methylation information. So if you take the second row, the first 4 cell types are all not methylated in these CpGs. The 5th one is methylated and the last one's not. Does that make sense? So you're finding the methylation or the CpGs in the genome that tell you what are the biggest differences between the phenotypes that I'm interested in. And here are the phenotypes. There's 6 different blood cell types. And this was identified using statistics, basic statistics. Okay, so another interesting question. So what about neighboring CpGs? So if you have one CpG in the genome that you found is differentially methylated, is a CpG right next to it also maybe going to be methylated or not methylated if it's close in a genomic sense? So many people have thought about this question, and there are 2 approaches I would say that people take. The first is, can you find 2 or more runs of differentially methylated CpGs? So if I perform this test site by site by site by site, a Ttest, for example, and I find the CpGs that are differentially methylated at a p-value greater than 0.05 or less than 0.05. That's one approach. The problem is, in statistics, when you do multiple testing, so when you test hundreds of thousands of sites in the genome, on average, you're going to find differences, and that's around 5% of the time. And that's totally normal, because we, as statisticians, accept 5% of the time being wrong. That's what the 0.05 means. So rather than just accepting that, statisticians have come up with ways to deal with multiple testing that correct for the fact that you're doing so many different tests in the genome. And that whole area of statistics is called multiple testing, if you're interested in it. So another approach is, how about we take the raw CpG data in a given gene or region of the genome and smooth across the CpG values. So what do I mean by that? Oh, there's my. Hold on, one second. One second. My plot won't show up, so I'm just going to re-import it and see if that helps. It says I have to restart my computer, which I'm definitely not going to do. And if it doesn't, then I'll move on. Okay, here we go. Okay, so let's consider a region of the genome. This is chromosome 2. We are looking at the methylation status of many different CpGs along this chromosome 2. So each one of those columns is one CpG site, and the red and the blue dots correspond to cancer and normal samples. So the methylation status of cancer and normal samples. So instead of comparing individual sites and performing hypothesis testing at each individual site independently, you could consider the entire region of the genome and try to find what are called differentially methylated regions as opposed to differentially methylated CpGs. So I have overlaid two lines here, the red line and the blue line. I'm going to explain what those lines are in the next slide, so hold on. But I want to give you some intuition of where we're going with this first. So the idea is you want to use the smooth methylation status between the cancer and the normal samples to identify regions where the mean difference of those smooth lines is larger than some cutoff or threshold. So that's what this second plot is. This represents the mean difference between these two lines. And when the mean difference is larger than some threshold, here it's 0.1 and -1, then you say that is a region that is differentially methylated. Does that make sense? Okay. So what is that? Where are the smooth lines? So I'm going to play a GIF to help you explain what it is. Okay. So the lines I mentioned before are created using what's called LOS, or locally weighted scatter plot smoothing. I'm not going to really explain what that is, but the GIF kind of tells you what's happening. Generally, whenever you have data measured over space or over time or distance, you should smooth because it's probably going to be fairly noisy. So this is one of the oldest ideas in statistics. It goes back, I think, regression from Galton, maybe, a long time ago. And the idea of LOS is you want to take small subsets of the data and apply many different regression models and generate this smooth value. So I can discuss more about that if you want, but I'm just going to leave it at that. Okay, so let's consider another example. Here we're considering 3 normal tissues: brain, liver, and spleen. Brain is in gray, that's this bottom line. Spleen is in purple, and liver is in pink. So the top plot illustrates the smooth and the unsmooth versions of the methylation values that we've obtained from our three different samples. And what's happening is when we smooth, similar to the blood type, we find that tissues also have a unique methylation profile. So we're able to identify tissues just based on their methylation status. So if you think about it, a heart cell and a lung cell contain the same information. What's different about them? The genes that are being expressed in the methylation status. So there's a huge push in genomics right now to try and identify these regions, these interesting genomic regions, people often call them. So that's some intuition behind that. Okay, the last topic. Really big in my field. It's often confused, but it's related to correlation and causation. And this is an XKCD that I love. It says, I used to think correlation implies causation. Then it says, then I took a statistics class, now I don't. And then this person says, sounds like it helped. And then the guy says, well, maybe. So, okay, so say you did an experiment, you obtained the data, you analyzed the data, you found an exciting result. Yay! Okay, so the first thing that should come to your mind is, what are any other explanations unrelated to my primary interest that could be causing this? Because what often happens is you have something else that's driving the difference that you're seeing. These are called confounders in statistics, and they are a huge pain in the butt, like huge. If you are a researcher and you're doing a project, be aware that confounders are a problem. So just because something is correlated does not mean that it's causal. That's the point of this. So what do I mean by confounding? Generally speaking, raw genomics data contains many biases and a lot of unwanted technical variations. So for example, the DNA methylation that you observe from one type of platform, so a type of a sequencer, one way of measuring it can differ than another way of measuring it, even though the true methylation status is the same. Just the process of putting it on a sequencer A and putting it on a sequencer B can drive a difference. Or say you ran a set of samples in the lab on Monday and then you ran a second set of samples on Tuesday. You could observe differences even though they're technically the same methylation. The true methylation is the same. That's unwanted technical variation that you don't want to deal with. These can cause perceived differences in your sample, irrespective of the biological variation. And then these differences are often due to experimental conditions that are hard or impossible to control for unless you're very careful in setting up your statistics or your experiments. And confusing them with biological variability can lead to false discoveries. So finding CpGs that are differentially methylated, that's an example of a false discovery in downstream analysis. Let me give you an example. Here are 57 samples from a bladder cancer study. And the goal of the study was to find differences between the normal and the cancer samples. And this plot is called a principal component plot, or the first two PCs. But the main point is you want the red points to cluster and you want the blue points to cluster. If the red points cluster, which they are the cancer samples, and the blue points cluster, that means you have enough genetic difference that you can actually find interesting differences. Okay, so this seemed like an exciting result because upon first glance, the cancer samples are all on the right side and the blue samples are on the left side. But what if I told you that these samples were processed on 5 different days or in 5 different batches? So an experimenter goes in the lab on Monday, processes a set of samples. They go into a lab on Tuesday, they process another set of samples. Wednesday, Thursday, Friday. You have 5 batches. Okay? This is all for the same experiment, but you just don't want to do them all in one day. Okay, so hopefully the fact that you process the samples on 5 different days has no impact on the variation that you see here, right? So now we're going to consider the same plot, but instead of coloring by the cancer normal status, we're going to color by the batch. So, for example, the day that the samples were processed. The problem is you can see that the samples group based on what batch they were in. That's a problem because it's hard to tell if the differences that you see are due to the cancer normal status or if this batch problem is driving the differentiation that you see. So this is an example of unwanted technical variation that I found very convincing when I saw it. There are even worse cases than this, but I thought this one was interesting. Okay, so how do you take this data that contains batch effects, that's noisy, and process it? I'm going to skip on all the standard normalization. I'm just going to cut right to one that most people use. It's called quantile normalization. It's by far the most widely one used for multi-sample normalization, so having 2 or more samples, not just one sample. It was originally developed for gene expression microarrays, if you know what those are, but it's now applied to all areas of genomics. Chip sequencing, RNA sequencing, brain imaging, all over. It can be very helpful, very, very helpful in eliminating unwanted technical variation. But the problem is it's based on a really big assumption. And if your data doesn't meet that assumption, it can wash away that interesting variation. So you did all that work to find differences between cancer and normal samples. You apply quantile normalization, and then all of a sudden, all of the differences that you want to find are washed away. They've disappeared because of simply the way you normalize the data. Because quantile normalization is based on this really big assumption. Okay, how does it work? There are 4 squares here, rectangles, and they're all the same except I am describing the process of quantile normalization. So consider the first one. Take the rows, which represent the raw data, so DNA methylation level for CpG1, CpG2, CpG3, all the way down to hundreds of thousands of CpGs. And the columns represent the samples. So sample 1 with all of the methylation levels for all the different CpGs. and then column 2, which is sample 2 with all the methylation levels for CPG in there, and so forth. Okay, so to apply quantile normalization, it's basically 3 steps. You order the values inside of each column, average across the rows, and substitute the value with the average, then reorder the average values in the original order. That's it. That's quantile normalization. I can tell you what it does. It makes, if you have 450,000 values, it makes the distribution of those values the same across all the samples. That's what quantile normalization does. So it forces the distribution for each sample to be the same, and it works very well in practice. Whenever you have samples that do not have global differences between them. So if the distributions are generally the same and you just have a lot of technical variation, you want to remove that technical variation, quantile normalization will do it. But if you have two sets of distributions that look very different from each other, quantile normalization makes those distributions the same. So what do I mean by global differences? I think this is the most convincing plot I found. This is gene expression. I apologize for not sticking with DNA methylation, but it was so convincing, I just wanted to use it. This is gene expression of brain and liver tissues. So here are, instead of just considering the expression of 1 gene or 2 genes or 3 genes, I'm considering the expression of 20,000 genes for one sample. That's one line. So another line is 20,000 genes for sample 2. Another line are 20,000 genes for sample 3. But the differences are the red are liver samples and the green are brain samples. So the important thing to notice is that the distributions between these two groups are very different. So the liver tissues, for example, are much more peaked and sharp, while the brain tissues have a fatter tail and are more spread out. These are global differences between brain and liver tissues. That means that the genes that are being expressed in liver tissues are different than the genes being expressed in liver tissues. So here's a case in which quantile normalization would fail miserably because it would make all of those distributions the exact same, washing away that's the biological variation that you're after. Okay, so let's go back to our DNA methylation. This is the distributions of the 36 samples of the 6 different pure cell types. So I'm plotting all the methylation values for one sample. That's one line. And then all the methylation values for the second sample. That's another line. The colors represent the 6 different cell types. And the question is, should we apply quantile normalization to this data? Do you see global differences between the distributions of these lines? Like, are the lines different enough that you would be concerned? That's the question a lot of researchers face. Like, it's not always as clear whenever you have this. This is rare. This is very rare. This is often what you see. So another way of looking at it is box plots. I'm a big advocate of exploratory data analysis, and that's fancy word for histograms, scatter plots, basic tools that you can use to explore the data. So box plots are another one of my favorite plots to use. But here I'm showing you the box plots of the 36 different samples, and you could ask the same question: are there global differences between these samples? Maybe the red samples are a little bit more in than the rest, or are there shifts in the averages? I mean, you could go all day with this. So if you apply quantile normalization, I just wanted to give you a brief example of what it looks like. Again, it makes the distributions the exact same. Like, I could plot the densities, I could plot these lines, but it would just be one line because they would all be on top of each other. Does that make sense? The distributions become the same. So how can we quantify if there are global differences in our data? Are the cell types for the red ones different enough than the rest, and so forth? So I came up with a test called Quantro. It tests for exactly this thing. It tests for global changes between groups of data. It's an R package that you can use. I'm not sure if you know what R is, but it's the language that almost all statisticians use. If you're doing any kind of statistical analysis with biological data, I encourage you to check out something called Bioconductor. Bioconductor is a fantastic resource that contains, I must see like thousands of packages for data analysis of genomic data. But this package, it tests the assumptions of quantum normalization. It tests if there are global differences in your data. That's essentially what it does. And the main idea is you want to compare the variability within groups to the variability between groups. And if that sounds familiar, it is familiar because it's essentially an ANOVA. An ANOVA compares the variability between groups to the variability within groups. But instead of doing it on an observation level, I'm doing it on the distributional level. So I'm comparing the 36 distributions here, or I guess these two sets of distributions, and asking, is the variability that I see inside the liver tissue and inside the brain tissue larger than the variability across the tissues? And in this case, the variability across the tissues is much larger. Does that make sense? Okay, so we did this to a lot of different data to explore its potential. And I find this plot helpful in like determining when you should be using quantum normalization. So in the first figure, figure A, we are looking at gene expression data of a SNP. So we're looking at one particular spot in the genome, and there are 3 different groups of samples. And the first group of samples all have the GG base at that particular location. The second set of samples have the AG base, and then the third set of samples have AA. And essentially what you see is that the variability across and between these groups is really, really small. They're very clean. There's not a lot of technical variation, and there are no global changes between the groups. So quantum normalization would remove any noise that you see. Maybe like this one line is slightly different. It would make those lines on top of each other, making the distributions the same, but it's not necessary. The second case is whenever you're comparing the gene expression of 3 different groups, non-smokers, smokers, and asthmatics. And what you see is those distributions are much more variable across or within groups. So it's very hard to see if there are any differences across groups because the variability within groups is so bad. Does that make sense? In this case, there's a large amount of variability or batch effects within groups and no global changes. Therefore, quantum normalization is exactly what you need to remove that technical variation so you can do the next step of downstream analysis. And then the third plot is the one I've already shown you, in which you have a small variability within groups, but a large variability across groups, and the cross groups are brain and liver tissues. So here, I just want to point out, these global differences could be caused by 2 different things. One, the biological variations, which I'm assuming that's the case, the brain and the liver tissue, because I've colored it based on tissue. 2 would be global technical variability. So if for some reason, all of the liver tissues were processed on one day, and all of the brain tissues were processed on another day, and that caused the difference, that could also be detected by Quantro. You could have global differences detected based on technical variation and biological variation. But there is no way to determine if it's being caused by technical variation or not. It will just detect if there are global differences or not. If they are due to technical variation, of course you want to quantum normalize. If they're due to biological variation, you don't. And in this case, I know they're biological because I know what the samples are. They're liver and brain tissues. So I'm not going to quantum normalize. So final thought, statistics matter. It's really important in the analysis of any data in genomics and in the larger data analysis community. Statistics can help identify relevant biological variations. So for example, finding differences in CPG sites, smoothing across genomic regions, and then statistics can eliminate unwanted technical variation in your data. So for example, batch effects, which are very scary. And then my acknowledgments, this is my boss. He's currently doing a MOOC and he loves the Red Sox. So he recorded a video in a Red Sox hat and shirt. So that was really funny. And my funding and then I'll take questions.
We record these, so if you have a question,
So when you use the term global, like you say global, biological global, and then like technical global, is that the term? Well, or is it?
No, you're right.
So I guess what exactly do you mean by global?
So if you consider that example of brain and liver tissue, that is an example of global differences. So the distributions are very different from each other. If there were no global differences, the distributions would look almost the same. But the global part of it comes from the fact that there are maybe 70% of the genes that are different between the liver and the brain that causes the visual differences in the distribution. Does that make sense? So if you just imagine some large percentage of 20,000 genes is differentially expressed between brain and liver, say all of the genes in the liver tissue are just turned off and all the genes in the brain tissues are turned on for some reason, a large number of them, that could cause the shift that you see between the red and the green.
So it's not a term that you're using to say like a big difference in.
It is a big difference. This is a huge difference in gene expression data.
So for this, you would use the term
Global
differences. Okay, just the general term.
Yeah.
Thank you.
Sure. Thank you.
So whenever you're doing the Ttest to compare methylation rate to different CpG sites,
yes.
Did you assume that methylation rates beforehand were normally distributed, or did you have to do a test beforehand?
So that's a great question, and many people have written hundreds of papers. The Ttest is, I think, the most intuitive one. It was probably the first test used to identify CpGs that are differentially methylated. Now people are using much more complicated methods. So I agree with you that those assumptions may or may not have been met, but that was just the first approach that people used to find differentially methylated CpGs, which is very intuitive for undergraduates. For you to, if you're not familiar with DNA methylation or a Ttest, it's very intuitive. You're right. Technically you should always test commonality.
So can you explain to people who are here how you got involved in all of this? So from being an undergrad, what was it that you did here at LSU that made you want to go on and pursue a career in research?
Sure. So when I was an undergrad, I majored in, I started out, I think, as a chemical engineer, and then I realized the only part of chemical engineering I liked was the math. So I was like, okay, so I'm going to be a math major. So then I started in the math curriculum, and I did not want to teach high school. And then I got involved with a scholarship program called LSTEM and HHMI. They encouraged me to do the REUs, which led me down a research path. So I did an REU, or research experience for undergraduate. It's a great way to spend your summers doing something else entirely. So I did them in biostatistics, even though I was majoring in math, and I found I loved biostatistics. So then I decided to pursue a PhD in statistics. I went to Rice, partly because the Texas Medical Center, I think, is the largest medical center in the world, and I was extremely intrigued by that. So I went to Rice, I did my basic stat classes, and then they start you with biological research right away. I loved it, finished my PhD, and then I decided to do a postdoc, and I'm still deciding what the next steps are. That's my back.
So in the video you showed at the beginning with the Google executive, we talked about the analogy of left shoes and right shoes and how valuable it is to be the wearer of the shoes. So in the kind of research you do, what do you think is the biologists or people collecting data are the left shoes and statisticians are the right shoes? What do you think the proportion of those two is in research?
I don't think you have to be like a statistician to be able to do analysis, but it definitely helps to have a statistics background or take statistics courses to have the training to attack a problem with a right perspective. So if you were not familiar with the multiple testing problem, you can quickly get into deep water. So I first just want to clarify, you don't have to be a statistician to do what I do. Like you can just take stats courses and do cool biological research. But what is the left and right shoe? I think
The right shoes are just people who know what to do with their data.
Yeah. I think my job is definitely more rare than the left shoe people who do biology. I enjoy explaining to someone and providing clarity on how to do their analysis because it's very obvious whenever you talk to someone who's done some kind of analysis and they just feel helpless. Like it's always nice to see the light click on when they're like, oh, I understand now. This is how I'm going to show what I'm going to show. And there are not enough people doing what I'm doing. I'm by far the more rare person. Biology people, I have a great respect for them too. I can't convince myself to work in a lab all day and pipette. So,
I have to say, just what you said, there are not enough people that do what you do because we have that problem in our grant. We have a Biostatistics 4, and yet our PIs of our grant don't know how to take advantage of getting someone to look at their data and tell them what they're seeing.
And I would say, like, if you're doing an experiment, get a statistician in there early because it is a lot easier for a statistician to guide you to do the right experiment than to salvage an experiment that is, wasted. I encourage you, if you are ever in a biology lab or doing any kind of biological research, talk to a statistician before you do the experiment because they are your friend.
Do you have any more questions?
Had another question.
Sure.
For the quantile normalization thing,
Yeah.
Is that valid for non-parametric data as well, or does it only work with data that you can assume has?
Quantile normalization is non-parametric. There's no model assumed for quantile normalization.
Okay, thanks.
All right. Well, let's thank Dr. Hicks one more time.